Genetic disorders, a comprehensive overview. Welcome, aspiring physicians, to our deep exploration into the world of pathology. Today, we open a chapter that delves into the very blueprint of life, the human genome, and the consequences when that blueprint contains an error. We are about to enter the realm of genetic disorders. Genes and human diseases. Let us begin by addressing a common misconception. Genetic disorders are not rare, esoteric conditions confined to specialized textbooks. On the contrary, they are far more common than is widely appreciated. If we consider the entire human lifespan, the frequency of genetic diseases is estimated to be a staggering 670 per 1,000 individuals. More than half of the population will, at some point, be affected by a condition with a genetic component. The genetic diseases we encounter in medical practice represent only the tip of a vast iceberg. These are cases with genotypic errors mild enough to permit full embryonic development and live birth. Beneath the surface lies a much larger story. It is estimated that 50% of spontaneous abortions during early gestation have demonstrable chromosomal abnormalities. Consider these statistics: about 1% of all newborn infants possess a gross chromosomal abnormality visible under a microscope. Approximately 5% of individuals younger than 25 years will develop a serious disease with a significant genetic component. Human genetic disorders can be broadly classified into three main categories, each with distinct characteristics. First, we have disorders related to mutations in single genes with large effects. These mutations are powerful. They either directly cause the disease or create a very strong predisposition to it. Such mutations and their associated disorders are highly penetrant, meaning the presence of the mutation is associated with disease in a very large proportion of individuals who carry it. Because these diseases are caused by mutations in a single gene, they usually follow classic Mendelian inheritance patterns and are often referred to as Mendelian disorders. Though profoundly instructive, these disorders are generally rare, unless maintained in a population by powerful selective forces. The classic example is sickle cell anemia, which persists at high rates in areas where malaria is endemic. The second major category is chromosomal disorders. These arise from structural or numerical alterations in autosomes and sex chromosomes. We are not talking about single base changes here, but rather gains, losses, or rearrangements of entire chromosomes or large segments. Like monogenic diseases, these are relatively uncommon but associated with very high penetrance. The effects are often profound and widespread. Finally, we arrive at the third and most common category, complex multigenic disorders. These conditions are caused by intricate interactions between multiple variant forms of genes and environmental factors. The variations in these genes are not rare mutations, but common polymorphisms. Each polymorphism confers only a small increase in disease risk. No single susceptibility gene is necessary or sufficient to produce the disease. It is only when an individual inherits a specific combination of several such polymorphisms that the disease occurs. This category includes atherosclerosis, diabetes mellitus, hypertension, and autoimmune diseases. Before proceeding, let us clarify commonly used terms: hereditary, familial, and congenital. A hereditary disorder is derived from one's parents and transmitted through the germline across generations. Familial simply means a condition runs in a family. It might be hereditary or due to shared environmental exposures. Congenital simply means born with a temporal definition, not causal. For instance, congenital syphilis is present at birth but caused by infection, not a genetic defect. Importantly, not all genetic diseases are congenital. Individuals with Huntington disease inherit a devastating mutation at birth, but may not manifest symptoms until their 20s or later. Mutations. A mutation, in its most fundamental sense, is defined as a permanent change in DNA. The location of this change is paramount. Mutations affecting germ cells, sperm, and ova can be transmitted to progeny and give rise to inherited diseases. Mutations arising in somatic cells do not cause hereditary diseases, but are critically important in cancer genesis. Let us establish general principles relating to gene mutation effects. First, point mutations within coding sequences. A point mutation substitutes a single base in the genetic code, potentially leading to replacement of one amino acid by another in the final protein. These are termed missense mutations. Not all missense mutations are equal. If the substituted amino acid is biochemically similar to the original, it might cause little functional change, a conservative missense mutation. A non-conservative missense mutation replaces the normal amino acid with a biochemically different one. An excellent example is the sickle mutation affecting beta-globin. The nucleotide triplet CTC encoding glutamic acid changes to CAC encoding valine. This single amino acid substitution dramatically alters hemoglobin's physicochemical properties, giving rise to sickle cell anemia. A point mutation can have even more drastic effects by changing an amino acid codon into a stop codon called a nonsense mutation. This leads to premature termination of protein translation. The resulting short, truncated peptide is unstable and rapidly degraded, potentially causing severe anemia like beta thalassemia. Mutations within non-coding sequences can also be deleterious. Transcription is regulated by promoter and enhancer sequences. Point mutations or deletions involving these regulatory sequences can interfere with transcription, factor binding, leading to marked reduction or total lack of transcription. Point mutations within introns can lead to defective splicing, interfering with normal mRNA processing and resulting in failure to form mature mRNA. Deletions and insertions have different effects depending on whether they involve multiples of 3 base pairs. If 3 base pairs or multiples are involved, the reading frame remains intact, resulting in a protein lacking or gaining amino acids. A classic example is the common cystic fibrosis mutation, a 3 base deletion resulting in a protein lacking phenylalanine at position 508. However, if the number of affected bases is not a multiple of 3, the consequences are severe, causing frameshift mutations. The result is incorporation of incorrect amino acids followed by premature stop codons. Finally, we have trinucleotide repeat mutations characterized by amplification of 3 nucleotide sequences. Although the specific sequence differs among disorders, almost all share guanine and cytosine nucleotides. In Fragile X syndrome, there are 250 to 4,000 tandem repeats of CGG within the ephemerone gene's regulatory region. In normal populations, these repeats average around 29. Such dramatic expansions prevent normal gene expression, causing intellectual disability. These mutations are dynamic. The amplification degree can increase during gametogenesis between generations. Mutations can interfere with gene expression at various levels. Transcription may be suppressed by deletions and promoter mutations. Abnormal mRNA processing may result from mutations affecting introns. Translation can be prematurely halted by nonsense mutations, and some mutations may lead to expression of normal amounts of dysfunctional protein. Mendelian disorders. Virtually all Mendelian disorders result from single gene mutations with large, discernible effects. Every individual is estimated to carry several deleterious genes. Most are recessive and don't produce serious effects because we have normal, functional copies to compensate. About 80% to 85% of these mutations are familial, the remainder represent new mutations acquired de novo. Most autosomal gene mutations produce partial expression in heterozygotes and full expression in homozygotes. Sickle cell anemia perfectly illustrates this. When homozygous for the mutant gene, all hemoglobin is abnormal HbS type, causing severe hemolytic anemia. In heterozygotes, only a proportion is HbS, the remainder is normal HbA. Red cell sickling occurs only under unusual circumstances like lowered oxygen tension. This heterozygous state is called sickle cell trait. A single mutant gene may lead to many different effects, pleiotropism. Conversely, mutations at different genetic loci may produce the same trait, genetic heterogeneity. Sickle cell anemia exemplifies pleiotropism. The single beta-globin mutation causes HBS formation, red cell, hemolysis, vascular logjams, and complications including splenic fibrosis, organ infarcts, and bone changes. Transmission patterns of single gene disorders. Single gene mutations typically follow three inheritance patterns: autosomal dominant, autosomal recessive, and X-linked. Autosomal dominant disorders. Autosomal dominant disorders are manifested in heterozygous states. An individual needs only one mutant allele copy to express disease. At least one parent is usually affected. Both sexes are affected equally and can transmit the condition. When an affected person marries an unaffected one, every child has a 50% chance of inheriting the disease. Some patients don't have affected parents due to new mutations in the egg or sperm. The proportion of patients with new mutations relates to the disease's effect on reproductive capability. If a disease markedly reduces reproductive fitness, most population cases would be expected to result from new mutations. Clinical features can be significantly modified by penetrance and expressivity variations. Some individuals inherit the mutant gene but are phenotypically normal in complete penetrance. If a trait is seen in all mutation carriers but expressed differently, this is variable expressivity. Neurofibromatosis type 1 manifestations can range from few café-au-lait spots to multiple disfiguring tumors and severe skeletal deformities. In many autosomal dominant conditions, age at onset is delayed, with symptoms not appearing until adulthood, as in Huntington disease. The molecular mechanisms depend on mutation nature and affected protein type. Enzyme proteins are generally not affected; instead, receptors and structural proteins are typically involved. Most mutations are loss of function mutations leading to reduced gene product production or dysfunctional proteins. Many autosomal dominant diseases fall into familiar patterns. One common pattern involves diseases disrupting metabolic pathway regulation subject to feedback inhibition. Membrane receptors, like the LDL receptor, provide prime examples. In familial hypercholesterolemia, 50% functional LDL receptor loss results in secondary blood cholesterol elevation, predisposing heterozygotes to premature atherosclerosis. Another pattern involves mutations in structural proteins like collagen. When genes encode multimeric protein subunits, mutant allele products can actively interfere with normal multimer assembly. The collagen molecule is a trimer requiring all three chains to be normal for proper assembly and stability. Even one mutant chain prevents normal collagen trimer formation, causing marked collagen deficiency. This mutant allele is called dominant negative. This effect is illustrated in osteogenesis imperfecta, characterized by severe collagen deficiency and brittle bones that fracture with minimal trauma. Less common are gain-of-function mutations, which can increase normal protein function or impart wholly new activities. Gain-of-function mutation disorders are almost always autosomal dominant. Huntington disease exemplifies this. A trinucleotide repeat mutation creates abnormal Huntington protein that is profoundly toxic to neurons. Autosomal recessive disorders. Autosomal recessive traits comprise the largest Mendelian disorder category. They occur only when both gene locus alleles are mutated. Key features include: the trait usually doesn't affect parents, but siblings may show disease. Siblings have 25% chance of having the trait. If the mutant gene has low population frequency, there's strong likelihood the affected individual results from consanguineous marriage. Several features distinguish autosomal recessive from dominant disorders. Expression tends to be more uniform, with complete penetrance common. Onset is frequently early in life. Although new mutations occur, they are rarely detected clinically because individuals with new mutations are asymptomatic heterozygotes. Several generations may pass before descendants mate with other heterozygotes and produce affected offspring. Importantly, many mutated genes in autosomal recessive disorders encode enzymes. In heterozygotes with one normal and one mutant allele, equal amounts of normal and defective enzyme are synthesized. Typically, biological systems' natural margin of safety ensures cells with half the usual enzyme complement function normally. Therefore, autosomal recessive disorders include almost all inborn errors of metabolism. X-linked disorders. All sex-linked disorders are practically X-linked, and almost all are recessive. Males are homozygous for X-linked mutant genes because the Y chromosome isn't homologous to the X chromosome. Thus, these disorders are fully expressed in males. Other X-linked recessive disorder features include affected males don't transmit disorders to sons, but all daughters are carriers. Heterozygous carrier women's sons have 50% chance of receiving the mutant gene and being affected. Heterozygous females usually don't express full phenotypic changes due to paired normal alleles on other X chromosomes. However, because of random X chromosome inactivation in every female cell ionization, females have variable proportions of cells with active mutant X chromosomes. Therefore, heterozygous females may express disorders partially. Glucosamine 6-phosphate dehydrogenase G6PD deficiency illustrates this. This X-linked enzyme deficiency, predisposing to red cell hemolysis with certain drugs, is expressed principally in males. In females, some red cells may derive from precursor cells where the normal allele was inactivated, making them susceptible to drug-induced hemolytic reactions, though usually less severely than homozygous males. Biochemical and molecular basis of single gene disorders. Mendelian disorders result from single gene alterations leading to abnormal protein formation or significant gene product output reduction. The mechanisms can be classified into 4 categories: enzyme defects and consequences, defects in membrane receptors and transport systems, alterations in non-enzyme protein structure, function or quantity, and mutations causing unusual drug reactions. Enzyme defects and their consequences. Mutations may result in enzyme synthesis with reduced activity or reduced amounts of normal enzyme. Either case causes metabolic blocks. This can lead to 3 major consequences. First, substrate accumulation. Depending on block site, this may include intermediate product accumulation before the block. This buildup can be toxic. In galactosemia, galactose 1-phosphate uridyltransferase deficiency leads to galactose accumulation, causing severe tissue damage to liver, eyes, and brain. Lysosomal storage diseases exemplify this, where excessive complex substrate accumulation within lysosomes causes catastrophic cellular dysfunction. Second, metabolic blocks can cause decreased end product amounts necessary for normal function. Melanin pigment deficiency may result from tyrosinase enzyme lack, necessary for melanin biosynthesis from tyrosine precursors, clinically resulting in albinism. Third, there may be failure to inactivate tissue-damaging substrates. This is exemplified by alpha-1 antitrypsin deficiency. Individuals with inherited serum alpha-1 antitrypsin deficiency cannot inactivate neutrophil elastase released in lungs. This unchecked enzyme activity leads to progressive lung alveolar elastin destruction, eventually causing pulmonary emphysema. Defects in receptors and transport systems. Familial hypercholesterolemia is caused by mutations in the LDL receptor gene, resulting in inadequate plasma LDL removal by the liver. Heterozygotes have 2 to 3-fold plasma cholesterol elevation from birth, leading to tendon cholesterol depositions, xanthomas, and premature atherosclerosis. Homozygotes have 5 to 6-fold cholesterol elevations with severe early vascular atherosclerosis. Myocardial infarction before age 20 is not uncommon. Cystic fibrosis involves defective chloride and bicarbonate ion transport systems in exocrine glands, sweat ducts, lungs, and pancreas. This impaired anion transport leads to serious lung and pancreatic injury. Alterations in non-enzyme proteins. Genetic defects in non-enzyme proteins often have widespread secondary effects, dramatically exemplified by sickle cell disease. The hemoglobinopathies are characterized by globin molecule structural defects. In contrast, thalassemias result from mutations affecting globin chain synthesis amounts associated with reduced amounts of structurally normal alpha-globin or beta-globin chains. Other examples include osteogenesis imperfecta collagen defect, hereditary spherocytosis red cell membrane spectrin defect, and muscular dystrophies dystrophin defects. Genetically determined adverse drug reactions. Certain genetically determined enzyme deficiencies are unmasked only after affected individuals are exposed to specific drugs. This area, called pharmacogenetics, is of considerable clinical importance. The classic example is G6PD deficiency-associated drug-induced injury. Under normal conditions, G6PD deficiency doesn't result in disease, but upon antimalarial drug primaquine administration, severe hemolytic anemia results. Disorders associated with structural protein defects. Several important diseases are caused by mutations in genes encoding structural proteins providing cellular and tissue shape, stability, and integrity. Marfan syndrome. Marfan syndrome is a connective tissue disorder manifested principally by dramatic skeletal, eye, and cardiovascular system changes. Prevalence is estimated at 1 in 5,000 individuals. Approximately 70% to 85% of cases are familial with autosomal dominant inheritance, the remainder are sporadic from new mutations. Marfan syndrome results from inherited defects in fibrillin-1, a large extracellular glycoprotein. 2. Fundamental mechanisms lead to clinical manifestations, loss of structural support in microfibril-rich connective tissue, and excessive transforming growth factor beta TGF-beta signaling activation. Fibrillin is the major microfibril component in extracellular matrix, providing critical scaffolds for tropoelastin deposition to form elastic fibers. Although widely distributed, microfibrils are particularly abundant in aorta, ligaments, and ciliary zonules supporting eye lenses. Coincidentally, the tissues most prominently affected in Marfan syndrome. Mutational analysis has revealed close to 1,000 distinct FB-NUN gene mutations. Most are missense mutations giving rise to abnormal fibrillin-1 protein that can act in dominant negative fashion, actively inhibiting normal fibrillin fiber polymerization. The second pathogenic mechanism involves TGF-beta bioavailability. Fibrillin-1 plays crucial roles in controlling TGF-beta bioavailability. Reduced or altered fibrillin-1 forms give rise to abnormal excessive TGF-beta activation because normal microfibrils sequester TGF-beta in inactive states. This excessive TGF-beta signaling has deleterious effects on vascular, smooth muscle development and increases metalloprotease activity. Clinical features: skeletal abnormalities are often most striking. Patients are unusually tall with exceptionally long extremities and long, tapering fingers and toes arachnodactyly. Joint ligaments are lax double-jointed. The head is commonly dolichocephalic with frontal eminence bossing. Spinal deformities include kyphosis or scoliosis. Chest deformities present as pectus excavatum or pigeon breast deformity. Ocular changes include bilateral lens subluxation or dislocation, ectopia lentis resulting from ciliary zonule weakening. This finding should immediately raise strong Marfan syndrome suspicion. However, cardiovascular lesions are the most life-threatening features. The two most serious lesions are mitral valve prolapse and ascending aorta dilation due to cystic median necrosis. Medial support loss results in progressive aortic valve ring and aortic root dilation, causing severe aortic incompetence. Media weakening also predisposes to intimal tears, initiating aortic dissections, catastrophic events where blood cleaves medial layers, creating false channels. The great majority of Marfan syndrome deaths are caused by aortic dissection ruptures. Ehlers-Danlos syndromes (EDS). The Ehlers-Danlos syndromes comprise clinically and genetically heterogeneous disorders resulting from mutations in genes encoding collagen itself or collagen-modifying enzymes. Collagen biosynthesis is complex and multi-step, and genetic errors can affect numerous structural collagen genes or enzymes necessary for post-translational modifications. Tissues rich in collagen: skin, Ligaments, joints are frequently involved in most EDS variants. Because abnormal collagen fibers lack adequate tensile strength, skin is often hyperextensible and joints hypermobile, permitting grotesque contortions. Most professional contortionists likely have Ehlers-Danlos syndromes. Skin is extraordinarily stretchable, but extremely fragile and easily bruised. Minor injuries can produce gaping defects, and surgical repair is difficult due to lack of normal tensile strength. Basic connective tissue defects can lead to serious internal complications including colon and large artery ruptures, vascular type ADS, ocular fragility with corneal rupture and retinal detachment, kyphoscoliosis type, and diaphragmatic hernia classic type. The kyphoscoliosis type is the most common autosomal recessive EDS form, resulting from lysyl hydroxylase gene mutations. Because hydroxylysine is essential for collagen fiber intermolecular cross-linking, enzyme deficiency results in collagen synthesis lacking normal structural stability. Disorders associated with receptor protein defects. Familial hypercholesterolemia. Familial hypercholesterolemia, FH, is most commonly caused by LDL receptor gene mutations, resulting in inadequate plasma LDL removal by the liver. FH is one of the most frequently occurring Mendelian disorders. To understand FH pathogenesis, we must review normal cholesterol metabolism. Approximately 70% of plasma LDL is cleared by the liver using sophisticated LDL receptor-mediated transport processes. LDL binds to cell surface receptors clustered in specialized plasma membrane regions called coated pits. After binding, coated pits containing receptor-bound LDL are internalized, forming coated vesicles that fuse with lysosomes. Inside lysosomes, LDL dissociates from receptors, which are recycled to surfaces. LDL molecules are enzymatically degraded apoproteins hydrolyzed to amino acids, cholesterol esters broken down to free cholesterol. This free cholesterol crosses lysosomal membranes entering cytoplasm for membrane synthesis and cholesterol homeostasis regulation. Released intracellular cholesterol affects four processes, suppresses cholesterol synthesis by inhibiting HMG-CoA reductase, activates enzymes favoring excess cholesterol esterification and storage, suppresses new LDL receptor synthesis, protecting cells from excessive cholesterol accumulation. In familial hypercholesterolemia, this system breaks down. FH heterozygotes possess only 50% of normal high-affinity LDL receptors. This transport defect impairs receptor-dependent LDL catabolism, increasing plasma LDL levels 2 to 3-fold. Homozygotes have virtually no normal LDL receptors, therefore much higher circulating LDL levels. The molecular genetics of FH is extremely complex. More than 2,000 different LDL receptor gene mutations have been identified, classified into 6 groups based on functional consequences. Lysosomal storage diseases. Lysosomes are key intracellular digestive system components, small membrane-bound organelles containing powerful hydrolytic enzymes functioning optimally in acidic environments. Inherited lysosomal enzyme deficiencies cause 2 major pathologic consequences. First, missing enzyme substrate catabolism remains incomplete, leading to partially degraded insoluble metabolite accumulation within lysosomes' primary accumulation. Second, there is tight linkage between autophagy and lysosomal function. Because of undigested macromolecule accumulation, lysosomes cannot process autophagic vacuole-delivered organelles at normal rates, leading to dysfunctional mitochondria persistence and secondary accumulation of other autophagic substrates. Approximately 70 different lysosomal storage diseases have been identified, typically divided into categories based on substrate and accumulated metabolite biochemical nature. Tay-Sachs disease. Tay-Sachs disease is the most common G-mTOR gangliosidosis form caused by beta-hexasaminidase enzyme deficiency resulting in inability to catabolize G-mTOR gangliosides. This disease is especially prevalent among Ashkenazic Jews, with 1 in 30 carrier rates reported. Hexasaminidase A is absent from virtually all affected individual tissues, so GM2 ganglioside accumulates in many tissues. However, neuronal involvement in central and autonomic nervous systems and retina dominates clinical pictures. Neurons are ballooned with cytoplasmic vacuoles representing markedly distended lysosomes filled with gangliosides. Affected infants appear normal at birth, but begin manifesting signs and symptoms at about 6 months. There is relentless motor and mental deterioration, leading to muscular flaccidity, blindness, and increasing dementia. Over 1 to 2 years, complete vegetative states are reached, followed by death at age 2 to 3 years. Gaucher disease. Gaucher disease refers to autosomal recessive disorders resulting from glucose ribosydase gene mutations, the most common lysosomal storage disorders. This enzyme defect causes glucose riboside accumulation, principally in phagocytes, but in some subtypes also in central nervous systems. 3 clinical subtypes exist. Type A chronic non-neuronopathic form accounts for 99% of cases. Glucose riboside storage limited to mononuclear phagocytes without brain involvement. Type Roman II acute neuronopathic is the infantile acute cerebral pattern dominated by progressive central nervous system involvement, leading to early death. Type Roman III is intermediate. Of immense clinical importance is the recently discovered link between Gaucher disease and Parkinson disease. Glucose ribosydase gene mutation is the most common known genetic risk factor for Parkinson disease development. Gaucher disease patients have 20-fold higher Parkinson disease risks, and 5% to 10% of all Parkinson disease patients have glucose ribosydase gene mutations. Glycogen storage diseases. Glycogenoses result from hereditary deficiencies of enzymes involved in glycogen synthesis or degradation. Depending on specific deficient enzyme normal tissue distribution, glycogen storage may be limited to few tissues, widespread, or systemic. From pathophysiologic perspectives, they divide into 3 major subgroups. Hepatic forms: the liver contains enzymes that synthesize glycogen for storage and break it down into free glucose released into blood to maintain glucose levels. Inherited hepatic enzyme deficiencies involved in glycogen degradation lead to liver glycogen accumulation, hepatomegaly, and blood glucose concentration reduction, hypoglycemia. Von Gierke disease exemplifies this. Myopathic forms: in skeletal muscles, glycogen is used predominantly as energy sources during physical activity. If glycolytic pathway enzymes are deficient, glycogen storage occurs in muscles, with muscular weakness due to impaired energy production. McArdle disease exemplifies this category. Some glycogen storage diseases don't fit hepatic or myopathic categories. Pompe disease involves lysosomal enzyme acid alpha-glucosidase deficiency leading to glycogen lysosomal storage in all organs. Cardiomegaly is the most prominent feature. Complex multigenic disorders. Complex multigenic disorders are caused by interactions between variant gene forms and environmental factors. The genetic variants involved are not rare high-impact mutations but common polymorphisms, each with modest effects and low penetrance, co-inherited by individuals. 2 additional facts have emerged from extensive complex disorder studies. First, while disorders result from collective polymorphism inheritance, different polymorphisms vary in significance. Second, some polymorphisms are common to multiple same-type diseases while others are disease-specific. Several normal phenotypic characteristics are also governed by multifactorial inheritance. Hair color, eye color, skin color, height, intelligence show continuous population variations producing standard bell-shaped distribution curves. Environmental influences can significantly modify complex trait phenotypic expressions. Chromosomal disorders. Normal human somatic cells contain 46 chromosomes, 22 homologous autosome pairs, and 2 sex chromosomes. Karyotyping chromosome study is the basic cytogeneticist tool. The aberrations underlying cytogenetic disorders can be abnormal chromosome numbers or structural chromosome alterations. Aneuploidy refers to cell acquisition of chromosome complements, not exact multiples of haploid number 23. Usual aneuploidy causes are non-disjunction, chromosomes failing to separate properly during cell division, and anaphase lag chromosomes left behind during division. Abnormal gamete fertilization by normal ones results in trisomic Twan+1 or monosomic Twan1 zygotes. Structural chromosome changes usually result from breakage followed by material loss or rearrangement, including deletions, ring chromosomes inversions, isochromosome formation, and translocations. Trisomy 21 Down syndrome. Down syndrome is the most common chromosomal disorder and major intellectual disability cause. U.S. newborn incidence is about 1 in 700. Approximately 95% of affected individuals have trisomy 21 chromosome counts of 47 with 3 chromosome 21 copies instead of 2. Maternal age strongly influences trisomy 21 incidence, occurring once in 1550 live births in women under 20, contrasting with 1 in 25 for mothers older than 45. Diagnostic clinical features are usually readily evident at birth, including flat facial profiles, oblique palpebral fissures, and epicanthic folds. Down syndrome is a leading severe intellectual disability cause. Approximately 40% of patients have congenital heart disease. Children with trisomy 21 have high leukemia development risks. Virtually all trisomy 21 patients living past age 40 develop Alzheimer's disease neuropathologic changes characteristic. Despite these problems, improved medical care has dramatically increased trisomy 21 individual longevity. Currently, median death age is 47 years. Sex chromosome disorders. Genetic diseases associated with sex chromosome changes are far more common than autosomal aberration-related ones. Sex chromosome imbalances are much better tolerated than similar autosomal imbalances, largely relating to lionization, X chromosome inactivation in female cells, and modest Y chromosome genetic material amounts. Klinefelter syndrome is defined as male hypogonadism occurring with 2 or more X chromosomes and 1 or more Y chromosomes. Incidence is approximately 1 in 660 live male births. Most patients have distinctive body habitus with elongated bodies, unichoid habitus with abnormally long legs, small atrophic testes, and lack of secondary male characteristics. Turner syndrome results from complete or partial X chromosome monosomy, characterized by hypogonadism in phenotypic females, affecting about 1 in 2,000 live-born females. Principal clinical features include significant short stature and primary amenorrhea. At puberty, there's failure to develop normal secondary sex characteristics. Single gene disorders with non-classic inheritance, trinucleotide repeat diseases. Fragile X syndrome is the most common genetic intellectual disability cause in males. It results from trinucleotide expansion mutations in ephemeron genes. Males with Fragile X syndrome have marked intellectual disability and often characteristic physical phenotypes, including long faces with large mandibles, large everted ears, and macroorchidism. The molecular basis lies in ephemeron genes characterized by multiple CGG sequence tandem repeats in 5 untranslated regions. Normal populations have small repeat numbers 6-55. Patients with full Fragile X syndrome have extremely large repeat region expansions 200-4000 repeats. Mitochondrial gene mutations. Several crucial mitochondrial genes exist that are inherited differently from nuclear chromosomal genes. Mitochondrial DNA features unique maternal inheritance. Ova contain numerous mitochondria, while spermatozoa contain few. Hence, mtDNA complements derive entirely from ova. Leber hereditary optic neuropathy is a mitochondrial disorder prototype, a neurodegenerative disease manifesting as progressive bilateral central vision loss first noted between ages 15-35, eventually leading to blindness. Genomic imprinting. Important functional differences exist between maternal and paternal alleles for some genes, resulting from imprinting epigenetic processes. Imprinting selectively inactivates either maternal or paternal alleles. Prader-Willi syndrome is characterized by intellectual disability, short stature, hypotonia, profound hyperphagia leading to obesity and hypogonadism. In 65% to 70% of cases, chromosome 15 long-arm specific band interstitial deletions can be detected, affecting paternally derived chromosome 15. In contrast, Angelman syndrome patients have deletions of the same chromosomal region but derived from mothers. Patients have intellectual disability, microcephaly, ataxic gaits, seizures, and inappropriate paroxysmal laughter. Molecular genetic diagnosis. Molecular genetic diagnostics has undergone breathtaking evolution from labor-intensive, low-throughput methods to next-generation sequencing NGS capabilities. NGS has continued improving dramatically in speed and cost, now possible to sequence entire human genomes for under $1,000 in days. When considering molecular genetic testing, genetic markers can be constitutional, present in every affected person's cell, or somatic, restricted to specific tissue types or lesions. PCR polymerase chain reaction has been molecular diagnostics' mainstay for decades. PCR involves DNA template synthesis of relatively short DNA fragments. Target DNA sequences between designed primer sites are exponentially amplified from single original copies. FISH fluorescence in situ hybridization uses DNA probes recognizing specific chromosomal region sequences. These probes are labeled with fluorescent dyes and applied to metaphase chromosome preparations or interphase nuclei. NGS applications range from targeted gene panels to whole genome sequencing. Each NGS analysis generates staggering sequence data amounts requiring specialized bioinformatics training for analysis. Because NGS can detect genetic anomalies of essentially any size scale, from single nucleotide polymorphisms to large rearrangements and aneuploidy, almost all today's genetic diagnostic test modalities could be supplanted by NGS in the future. We have journeyed through genetic disease landscapes from subtle single nucleotide errors to profound entire chromosome imbalances. This knowledge is not static but a vibrant, rapidly evolving field at modern medicine's heart. As you continue studies and future careers, you will witness how our deepening human genome understanding continues transforming how we understand, diagnose, and treat human disease.